कर रहा है? Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on Earth, and that's biology. So thank you so much for joining. Well, let's discuss now series. So here we are discussing the first sample paper. So we are towards the end of this sample paper. We are going to discuss the last 25 questions of this sample paper. So this is second part of zoology, I would say. So after this, we'll be starting with a new sample paper. So in between, I have planned something all the more again interesting as well. So let's see how it goes out. So just 25 questions as a part of zoology. So when we saw zoology part 1, so it felt much more easier, right? So I hope this part is also easy. So let's see how easy it is. So here we go. So first question, which of the following factors affect 𝗍𝐴𝐴𝐵 equilibrium? So which are the factors? Genetic drift, gene flow, mutation, all of these. So which of them? So this question comes from the chapter evolution, right? So this comes from the chapter evolution. So what is 𝗍𝐴𝐴𝐵 equilibrium? So in short, if you want me to say, it's p square plus q square plus 2pq is equal to 1. So when the genetic equilibrium is maintained, so the answer is all of these. So when there is a genetic drift or a gene flow from one group of population to the other, when there is random mutation, so all this affects your 𝗍𝐴𝐴𝐵 equation. So in that case, it will not be equal to 1. All right, so this is question 1. So question 2, C-onc genes are found in. So C stands for cellular. Onc stands for oncogenes. Okay, so these are found in. This question comes from Human Health and Diseases. So cellular oncogenes are part of the normal cells which under certain circumstances become your oncogenes, that is, they become cancerous. So cellular oncogenes are proto-oncogenes. They are in a stage before getting activated into cancer genes. Alright, so question 2 is done. So we go to question 3. So SMAC is the common name of heroin. So this comes under the drug abuse. So from the chapter Human Health and Disease again. Okay, so I go to question number 4. The best practice of mating animals within the same breed. But there is nothing called as inbreeding here, right? So when you mate within the same breed, that is inbreeding, right? But there's a condition there for inbreeding. So inbreeding is that the generations are for 3 to 4 generations. You mate within the same breed for 3 to 4 generations. But here they have not mentioned any generation. So it is a type of outcrossing, I would say. Yes. So outcrossing is done to overcome inbreeding depression, if you remember. So this is, they still mate within the same breed who do not have common ancestors. So this question comes from this chapter, Strategies for Enhanced Food Production. Okay, so here we go to the next question, question 5. So penicillin as an effective antibiotic was commercially extracted by which group of scientists? Ernst Chain and Howard Florey. Okay, though these two names are not given in your textbook other than Alexander Fleming, who discovered your penicillium. So it is these two scientists, Ernst Chain and Howard Florey. But so the answer is both B and C. But questions of these type rarely come, so there's a less possibility of scientist name coming other than the names that is given in your textbook. Okay, so this comes from the chapter again, Human Health and Disease. So question 16, which of the following acts as a blood cholesterol lowering agent? So this comes under the topic microbes in human welfare. The last chapter of unit 3, so this is statins. The statins act as blood cholesterol lowering agent, inhibiting the cholesterol synthesizing enzyme, right? So this statin is produced by Monascus purpuris. So question 7, in a biolistic method of gene transfer. So when you talk about gene transfer, the chapter is biotechnology. right? The cells are bombarded with high velocity microparticles. The microparticles used are tungsten and gold, if you remember. So it is both A and C. All right. So this chapter is very important when you consider class 12. So chilled ethanol is added during DNA isolation because it precipitates the purified DNA. And this is again from the same topic, biotechnology, principles and process. So question 9, RNA interference includes silencing of an mRNA, right? It's a type of gene silencing mechanism and it's a type of defense mechanism in eukaryotes. So this comes from biotechnology and its applications. Okay, so here we move forward. So question 10. The separated bands of DNA are cut out and extracted from the agarose gel by the process called. So after I have found which is my gene of interest on the agarose gel, so imagine this is my ladder and I'm getting bands like this. So if this is my gene of interest, I have found it with reference to my ladder. I have to take this back so that my restriction ligation can be done. So this process of taking the DNA from the gel is called as elution. This is option B. So this again comes from the same topic, that is biotechnology, principles and process. So question 11. So you have to do the match the following. So this match the following, even if we are not very clear about which example comes under whom. So simplest method is deuteromyces. So deuteromyces is imperfect fungi that all of us know. It's very easy to remember because they do not reproduce by the sexual way, right? So D4, where it is? The right answer should be B. So isn't it easy? So match the followings are in a way depends on a luck. It can be either easy or it can be very confusing, right? So here we go to the next question. So question 12. Yeah. Refer to the given figure showing arrangement of the germinal layer. So you have the 3 layers and identify the organism with the correct characteristic feature. So they are saying B could be Ascaris, in which the males are longer than the females. No, in roundworms the females are much longer than the males. Okay, so this is incorrect. A could be Pleurobrachia, which have comb plates for respiration. So comb plates are not used for respiration, though Pleurobrachia comes under Ctenophora. This is again incorrect. So A could be Balagnoglossus, having proboscis gland for excretion. So A is diploblastic. It has undifferentiated mesoderm, that is mesoglia. So Balagnoglossus are triploblastic. So again this is wrong. A could be Anopheles, having a chitinous exoskeleton. That is true because this comes under orthopods. So they have this mesoglial method. They have the well differentiated mesoderm. So B could be Anopheles. So yes. So it has ectoderm, endoderm and mesoderm. So this could be Anopheles, is a mosquito that comes under phylum Orthopoda. So this is the right answer. The question, so that question was from the chapter Animal Kingdom. So I hope you are able to recollect. The question 13, so you have to just match. It's very easy. So you have to fill in the blanks, okay? Connective tissue connects different tissues or organs of the body, right? Dash and dash are loose connective tissues. So connective tissue has a classification: loose connective tissue, dense connective tissue. So loose is your areolar and your adipose. So I write it down. Okay, which has cells and fibers loosely arranged in a semi-fluid ground substance. The dash tissue in this is the most widely distributed connective tissue, that is your areolar connective tissue, right? So our 1 is right, so this is matching. It is present under the skin. So the right answer is option A. So this comes under this chapter, structural organization in animals. So where you read about the animal tissues and about cockroach. So question 14, complete the following reaction and select the correct option. So starch gets converted into maltose in the presence of salivary amylase. So this question comes from the chapter digestion and absorption. Okay, so here the pH is 6.8. Then lactose gets converted in the presence of lactase. It gets broken down into glucose and galactose. Fats is broken down into glycerides in the presence of lipases. Diglycerides gets broken down into monoglycerides. So what is the answer? Salivary amylase, galactose, lipase, monoglycerides. Exactly the right one. So question 15, so you have to arrange the order of respiration in the correct sequence. So first it starts with contraction. So if you've seen my breathing and exchange PPT, so we have the slide, right? So where inspiration starts. So it starts with the contraction. So the process starts with 3. Okay, once it starts, then the ribs and the sternum are pulled upward. Okay, so this is number 2. So when they pull upward, the pulmonary volume increases. So when the air increases, the pressure decreases. So when the pressure decreases, the air moves inside, right? So the right arrangement will be B. Right? That is what happens. So this is from the chapter Breathing and Exchange of Gas. So this is from your body fluids and circulation. Given below is a flow chart showing the division of vascular tissues. So you have to identify A, B, C, D. Okay. So vascular tissues are divided into blood and another component. So what is A you have to find. So we go with blood since we are very clear with blood in this chapter. So blood is divided into plasma and formed elements. So formed elements is your erythrocyte, leukocyte and your thrombocyte or your platelets. Your platelets are also known as thrombocytes, right? So your leukocyte is divided into A granulocyte, which is lymphocyte, and your monocyte. So granulocyte is BEN, if you remember, basophil, eosinophil and neutrophil. So now tell me which is right. So B is plasma, C is thrombocytes. Okay, so I don't think, okay, we go with the first one. C is plasma. No, C is not plasma. D is not monocyte. So this is ruled out. So B is plasma, right? But C is not eosinophil. This is ruled out. So A, okay, so vascular tissue, it is blood and lymph, right? So they are type of connective tissue. So C is thrombocytes and E is monocytes. This is absolutely right option. Okay. So question 17, you have to again do the matching. So this is from locomotion and movement. So amoeboid movement is seen in macrophages, ciliary movement is seen in your fallopian tube, muscular movement in your jaws, flagellar movement in your sperm. So A is 3, B is 1, right? So this is the right answer. So question 18, okay, so this is from neural control and coordination. So this is from here. Select the option that correctly identifies the following statement. A thin elastic membrane above the row of hair cells. So that is your tectorial membrane. Right? So this should be the answer. An aperture surrounded by the iris, that is your pupil, right? The sensory part of saccule and utricle is maculae. The projection is maculae. And aldehyde of vitamin A is retinal. So the right option is C. So question 19, so you have to read the statements and tell which is correct. So glucagon is a hypoglycemic hormone. This is wrong. Glucagon, which is secreted by the alpha cells of the islets of Langerhans by the pancreas. So this increases the blood sugar level. So it is hyperglycemic hormone. So A itself is wrong. Okay. Glucagon increases the cellular glucose uptake. No. If the cell takes up glucose, then the blood sugar level will not be high, right? So it inhibits the cellular uptake so that glucose is sugar level is more in the blood. So here both the statements are incorrect. So both statement 1 and 2 are incorrect. This is your answer. So coming to question 20, which among the following statement is not true regarding menstrual cycle? Alright, so this comes from human reproduction chapter. LH and FSH attain a peak in the middle of the cycle. True. LH surge induces the rupture of Graafian follicles. True. Right? Estrogen is essential for the maintenance of endometrium. This is wrong. So progesterone is essential for the maintenance of endometrium. Thus, this is the incorrect statement. So for a change, let's read the D1 also. Increases during the follicular phase. This is also a right statement. Right? So question 21. A woman who had a surgical sterilization want to conceive a child. So this is a type of barrier methods and assisted reproductive technologies. So this comes from the chapter Reproductive Health. Right? So which among the following assisted technologies can help her have a child? So when they do sterilization technique, the fallopian tube is cut off and tied. So directly you have to do intrauterine transfer. Directly in the implantation stage, it has to be put inside, right? So it will be option C. So 22, you have to fill this. Dash and dash are the two key concepts of Darwinian theory of evolution, branching descent and natural selection. The different species in a given habitat starting from a point and radiates to other habitats is your adaptive radiation. So it should be two. So this is branching descent. Second one is natural selection. This is adaptive radiation. And this is your Tyrannosaurus rex. So the right answer is option B, right? So we go to question 23, the last three questions. Select the mismatched pair. Haemophilus influenzae causes pneumonia. So this comes from microbes in human welfare. Microsporum canis causes filariasis. This is wrong. And this is the correct one then according to the question, because this is caused by which area worm? Salmonella typhi, typhoid, entamoeba, histolytica, amoebiasis is also right. So question 24, the following table summarizes the difference between primary sludge and activated sludge. So we have to find the incorrect pair. Okay. So primary sludge is formed during the primary sewage treatment. It is formed during the secondary sewage treatment, right? But we have to find the incorrect pair. It possesses flocks of decomposer microorganisms. This is wrong. Primary sludge does not produce flocks, or it does not contain flocks. Formation of primary sludge requires aeration. No. So 2 and 3 are wrong. Okay, so we haven't read fourth one also. Maximum decomposition occurs during the formation, so that is also wrong. So 2, 3 and 4 are incorrect matches. So coming to the last question for the evening. So identify the technique based on the given characteristic, right? So it is used to detect HIV. The moment you read this, you will end up in ELISA. But hold on, please read it fully. It is used to detect HIV in suspected AIDS patient. It can be used to detect mutation in genes suspected in cancer patients. So can ELISA do this? So it can be used to detect mutations. You're still not very sure. It can detect very low amounts of DNA. So now what is the answer? The right answer would be PCR. Okay, so PCR can be used to detect mutations. You can amplify a particular gene and look for mutation. Alright, yes. So Western blotting is to study RNA. Southern blotting is for DNA. Sorry, Northern blot is for RNA. Southern blot is for DNA. Western blot is to study the protein. Alright, yes. Okay, so with this we come to the end of this first sample paper. So I have discussed in 4 parts. I've discussed 25 questions in each session. So I hope you found it helpful and you are able to understand the question, analyze the question and solve along with me. So tomorrow I'll start with a new paper. So the second sample paper we'll start with, alright. So the first part, botany of that paper we'll be doing, alright. So I hope you enjoy the session. So please do like, share and subscribe. So I'll meet you tomorrow at 3:15. Yes, the timing has been preponed to 3:15 instead of 4:15. So please make a note in your schedule and I'll meet you tomorrow. So until then, this is me signing off. So take care and bye. Thank you.